Okay, now that we're done with passive transport, let's talk about active transport. So remember, passive transport does not require any additional energy. It's all due to differences in concentration and physics. And so think primary active transport. So again, ATP, that magical molecule, provides a lot of energy. So if it's active transport that uses ATP, it's primary active transport. But secondary active transport, instead of this is not independent of ATP. It does not use ATP, but rather it uses up the stored up potential energy from chemical gradients. Again, a difference in chemical concentrations between the outside and inside of a cell. So the thing is that endocytosis and endocytosis, these are also forms of active transport. They do use ATP. They are forms of primary active transport. But again, I could go down the rabbit hole with that, but I think I'm going to leave that for my colleagues in neuroscience, and especially with endocytosis and exocytosis, very important in vesicle trafficking and anterograde and retrograde flow. But again, these are the two, like primary and active, secondary active transport are the ones I want you to concentrate in this lecture. Okay, so primary active transport. Here is a classic example of primary active transport. If you haven't had questions on this during your undergrad career or on the MCAT, I guarantee this is probably going to show up in some way on the USMLE1, like the sodium potassium exchange pump. Very important. So things that, again, remember, ions and solutes are not in equal concentrations inside and outside of a cell. But what happens when you open up these ion channels that allow the free diffusions of these ions and solutes? Well, they're going to eventually reach an equilibrium. So how do you get them back to where they have that unequal concentrations, those concentration gradients between the inside and outside of a cell? So one thing that's very important, especially for excitable cells such as muscle cells and neurons, things that when they do something called action potentials, that basically undoes all the differences in chemical ion concentrations, especially with sodium, potassium, and in case of muscle cells, calcium as well, or actually neurons too, calcium as well. But basically, so how do you get them back to where there's more potassium on the inside, more sodium on the outside? So what we have here is the sodium potassium exchange pump. So pretend you did an action potential. And again, I'm showing you a very simplified version here. But what the sodium potassium exchange pump does is take 3 sodium ions. And again, a lot of these exam questions, they love using this, like not just like sodium and potassium moving, but how many ions are exchanged for how many ions. So you have 3 sodium being pumped out via ATP. So ATP is going to cause a change in the conformation and shape of the sodium potassium exchange pump. So it loads the 3 sodiums here from the inside of the cytoplasm, and then it changes shape. And this conformational change, now the exchange pump is going to release the sodium ions into the extracellular fluid. Why did this require energy? Well, for simplicity's sake, I didn't show you the myriad of sodium ions, probably, I don't even know how many trillions and billions, but probably more quadrillions, but there's a lot more sodium ions here on the outside. So things that if you're trying to push, it's kind of like, have you ever tried to have a dresser full of clothes? It's already full of clothes, and as your dresser gets full of more and more filled with clothes, do you have to use more effort to push more clothes in that dresser, or do you need less effort? Again, the more there's already in there, the harder it is to push more and more clothes in there. So same with sodium. There's so much sodium out there in the extracellular fluid, it takes energy to pump out sodium from the inside of the cell to the outside of the cell, just because there are so many sodium ions there already. Same with potassium ions. It takes energy to move that, but the thing is that you already have this change in shape. Now you can bring in 2 potassium ions, and by losing that phosphate group due to the phosphorylation, so by losing that, that causes it to revert back to its original state, and this releases potassium on the inside. So just repeat this ad nauseum. Now what do you have? You just repeat again, 3 sodium from the inside, exchanging from 2 potassium on the outside. So pump sodium out, bring potassium in. Now we have a higher concentration of sodium back on the outside and a higher concentration of potassium on the inside. Again, especially for neuroscience and for anything involving action potentials, whether it's skeletal muscle or any other muscle, cardiac muscle, or smooth muscle, and also neurons, especially neurons. This is a very important concept to know. Okay, so that was primary active transport because it involved ATP with that sodium potassium pump. Now let's talk about secondary active transport. So here we have sodium. Again, at rest, we have more sodium on the outside of a cell. And now we are talking about calcium. And there's a special protein, transport protein called the sodium calcium exchanger. So what the sodium calcium exchanger is, uses the concentration gradient, again, fancy term for difference, more sodium on the outside, and uses that flow from the natural tendency of sodium to flow from the outside to the inside, if given the opportunity, to drive the movement of calcium. So what it does is take 3 sodium ions, and then it takes the calcium from the inside of the cell, and due to the increased relatively large concentration of sodium versus the concentration of sodium in the cytoplasm, it uses that energy to drive calcium into the extracellular fluid. So this is also, there's also calcium pumps that require primary active transport to pump it into the extracellular fluid, but this is another way of moving calcium into the extracellular fluid. Now, in that previous example, let's play again. Did we see ATP involved at any point? And 3 sodium, and just due to more sodium being on the outside, that energy is driving calcium outside. We did not see ATP. This is why this is secondary active transport. It just relied on the potential energy from sodium being more concentrated on the outside versus the inside to drive calcium against this concentration gradient, drive it from, because again, remember, at rest, there's more calcium on the outside, so it's hard to push more calcium from the inside of the cell towards the outside of the cell. But by having more sodium, this is allowing it to provide the energy to drive calcium out. So I'd like to use this. So what happens if you have this repeating over and over again? Well, you're starting to pump more and more calcium out, and you're at the expense of allowing sodium to flow in. So I'd like to use this. I don't think the Circus has been around here for a while. I don't know if there are any circuses still traveling, but you might have seen these, like maybe like Cirque du Soleil or one of those things where the acrobats. You have an acrobat on a platform, and you have another acrobat on a seesaw down here. So what happens when the acrobat on the platform does that flippy dippy do stuff and jumps and hits that seesaw? Well, what's going to happen to the acrobat that was waiting down here? So that the acrobat that jumped from the platform, he went from all that potential energy transferred to kinetic energy. So he went from high to low energy, but that energy is going to be transferred into the seesaw and into that acrobat that was in wait. So that's similar to what happened with the sodium and calcium ions. That potential energy from all that sodium being built up on the extracellular fluid is being used as it goes inside or it flows into the cell via the sodium calcium exchanger. This energy is being used to drive calcium into the extracellular fluid. Again, this is secondary active transport because it did not use ATP. So again, that's what happened there. So we have secondary active transport. And again, is there any ATP being involved in this? Nope. So again, 3 sodium, 1 calcium, and this pumps calcium into the outside. And repeat it again, and then now you have calcium being pumped into the outside at the expense of bringing in sodium. Again, no ATP at any point. This is secondary active transport.